All right, diving in today, folks. We're looking at how cells manage all those crazy complex production lines they've got going on inside, their metabolic pathways.
Yeah, it gets pretty wild down there.
For sure. And our awesome listeners have pointed us to some really cool articles focusing on two big ways these cells keep everything running smoothly: feedback inhibition and feedback repression. So basically, how do these tiny factories maintain order and efficiency?
Exactly. And that's our mission today, right? To break down these vital control systems, show why they're so elegant, even if you're not like a hardcore science buff.
Totally. We're taking the research you guys sent and pulling out the good stuff, the need to know.
Absolutely. So, should we kick things off with feedback inhibition?
Yeah, let's jump right in. I think I remember us talking about a car factory analogy before, right? Like if they're churning out too many finished cars, it's got to be a good idea to pause things earlier on in the line, right?
Exactly. That's the basic idea behind feedback inhibition inside a cell.
Okay, that makes sense. But how does the cell actually, like, hit the brakes?
Well, it often uses something called allosteric irregulation. Think of it this way. An enzyme at the beginning of a pathway has two important spots.
Uh-huh.
So you've got the active site where it does its main job.
Right.
And then there's another spot called the allosteric site.
Okay.
And that's where the end product of the whole pathway acts like a regulator. It binds to this allosteric site.
Gotcha. So the end product actually physically changes the enzyme then?
Precisely. When it binds, it kind of forces a shape change in the enzyme. Imagine a key fitting into a different lock on the same door. And this new shape makes it tougher for the enzyme to connect with its usual starting material.
So it slows the whole thing down.
Exactly. Or it could just slow down the enzyme's activity in general. Either way, the result is the same.
Right.
Less of that end product gets made.
Brilliant. And the cool thing is it can change back too, right?
Yep, it's reversible.
Yeah,
And that's super important because it means the cell can respond quickly to what it needs.
that makes sense. So the research mentioned isoleucine biosynthesis as a good example, right?
Yes, perfect example. Threonine deaminase, that's the first enzyme needed to make isoleucine.
Right.
But isoleucine itself, the final product, also acts as the inhibitor for threonine deaminase. So you have this direct feedback loop, almost like a thermostat for isoleucine production.
I love that analogy. So simple.
Right. When there's enough isoleucine, it tells the cell to chill out on making more.
Perfect. Okay, so let's shift gears a bit and talk about feedback repression. This one sounds like it's more of a long-term strategy, right?
Yeah, you could say that. Instead of just putting the brakes on the existing enzymes, the cell actually reduces how many of those enzymes it makes in the first place.
Oh, I see. So going back to that factory analogy, it'd be like the boss saying, hold up, we've got way too many cars piling up. Let's make fewer machines that build engines.
100%.
Yeah.
It's like controlling the blueprints, right?
Exactly.
And this all happens at the level of gene transcription, which is basically how the cell creates a working copy of a gene. It does this using messenger RNA, or mRNA. And the process often uses a repressor protein that attaches itself to the cell's DNA. It's like a guard dog.
I like that.
And the end product, or sometimes a molecule related to it, usually acts as a co-repressor. They team up.
Okay, so when there's not enough of the end product, the repressor is like off duty. The genes that make the enzymes for the pathway can do their thing, but when there's tons of the end product,
And when there's a ton of the end product, it binds to the repressor, flips that switch, and activates it. Like, all right, guard dog, time to work. And now this activated repressor attaches itself to a specific region of the DNA called the operator,
okay.
which happens to be right next to the genes for the whole pathway. And by attaching there, it blocks RNA polymerase.
And what's that exactly?
RNA polymerase is the machinery that actually creates mRNA from DNA. So it's
Okay.
like putting up a roadblock right in front of the factory entrance.
That is wild. The tryptophan example in E. coli really shows this in action, right?
Oh yeah, great example. The tripoperon. That's the cluster of genes responsible for making tryptophan. And there's this protein, TRPR, which acts as the repressor.
Right.
And get this, tryptophan itself is the co-repressor.
So it controls its own production. That's nuts.
Right. So basically, when tryptophan levels are high, it activates the repressor, which then blocks the cell from making more of the enzymes that produce tryptophan.
Mhm.
It's like the cell has this built-in mechanism to say, hey, we've got plenty of tryptophan, no need to make more.
Wow, that's fascinating. So you've got these two awesome systems, feedback inhibition for those quick adjustments to how active the enzymes are, and then feedback repression to control the actual production of enzymes over the long haul.
Exactly. And these are just two ways cells can fine-tune things. I mean, there are likely others we haven't even discovered yet.
Yeah. So why are these systems so crucial? What's the big deal?
Well, they're all about maintaining balance inside the cell, something called homeostasis. They prevent resources from being wasted. It's like the
Hmm.
cell's way of making sure things only happen when they need to happen. Super efficient.
Right. And the articles you shared also talked about how scientists are figuring out how to manipulate these systems, right?
Oh, absolutely. Deregulation, messing with those control systems,
Mhm.
that's huge in industrial biotechnology.
Right.
Think about it. If you want to mass produce something super valuable, like, I don't know, some kind of primary metabolite,
Uh-huh.
you need to figure out ways to either weaken or completely bypass those natural feedback mechanisms. Some researchers are even looking for mutant organisms that naturally have less feedback control.
That's crazy. So in the end, we've got these two elegant solutions for cellular efficiency
Totally. And it really makes you wonder, right? If these natural systems are so good at regulating themselves, what happens when we humans try to override them?
for our own purposes.
Right, whether it's for industrial production or even in understanding like metabolic issues in our own bodies,
Right.
what other layers of regulation haven't we even uncovered yet? There's so much more to explore.
Food for thought, everyone. That's our deep dive for today. Thanks for joining us.
Until next time.